Good day, everyone. So we're reviewing Wetzel's Limnology textbooks. This is edition number 4, and we're on chapter 14, the nitrogen cycle. And more specifically, we are looking at nitrogen sources, transformations, and fate in streams and rivers, which is subchapter 3. And where we left it at yesterday is this subparagraph here, the water flow in streams. So water flow in streams and rivers can continuously bring dissolved nutrients to primary producers, especially attached ones. However, nutrient limitation in streams and rivers has been found in many studies and shows regional and seasonal variation. From a global review of nitrogen and phosphorus limitation on primary producers in freshwater, marine, and terrestrial ecosystems, the frequency of phosphorus limitation in freshwater systems, including streams and rivers, was as common as in marine and terrestrial systems. This global review also demonstrated that compared to other systems, marine systems have a higher response to nitrogen enrichment, but freshwater systems have a higher response to simultaneous nitrogen and phosphorus addition. Figure 14-11. Responses of autotrophs to single enrichment of nitrogen in red, or P blue phosphorus, or to combine nitrogen and phosphorus enrichment in purple. In terrestrial, freshwater, and marine ecosystems, data are given as natural log transformed response ratios, or RRX, and that is in the y-axis, in which autotroph biomass or production in the enriched treatment is divided by its value in the control treatment and then natural log transform. Thus, a value of 0.5 indicates a value in the manipulated treatment that is 1.6 times its value in the control, while a value of 1.0 indicates a 2.7-fold increase. Sample sizes of nitrogen, phosphorus, and nitrogen and phosphorus treatments were 112, 107, and 126 for terrestrial studies, 509, 506, and 618 for freshwater studies, so this is where we have the most samples, and 149, 141, and 197 for marine systems. So marine systems and terrestrial systems are about the same sample size, pretty close. Error bars indicates plus or minus 1 standard error. Okay, so terrestrial system. What we see here in terrestrial system, the best response is when we have a nitrogen and phosphorus combination treatment. We have about the same response if it's only nitrogen or phosphorus that is added. So they were saying that 0.6 is an increase of like, so 0.5 is an indicator of 1.6 times. So we have close to 0.5, so this is, like 1.6 times a response higher in the manipulated treatment compared to the control. Freshwater, definitely, we see a very good response with the nitrogen phosphorus combined. We have 1.2. And then pretty standard response in terms of if it's only nitrogen or phosphorus. So a little better response for phosphorus than nitrogen actually in the freshwater, not by much. And then nitrogen phosphorus, and then in the marine environment, for nitrogen phosphorus, the same leading component that we've seen for terrestrial and freshwater, where the response is optimized when we have nitrogen and phosphorus that is amended. And definitely the marine ecosystem is very responsive to just nitrogen being added compared to phosphorus. So phosphorus we have 0.2, and then nitrogen we have about 0.5 or something to that effect. So which means, once again, that is 1.6 times higher in response in the treatment than it is in the control. A global scale riverine nitrogen flux calculation estimated that approximately 20% of nitrogen from catchments was retained in rivers, streams, prior to reaching the coast. Headwater streams have been considered key sites to process and remove nutrients, thus reducing their downstream export. A large-scale study, including 123 tributaries in the Mississippi River Basin, showed that the first-order nitrogen loss rate decreased from 0.45 to 0.005 per day, with increased channel size. This study also concluded that the proximity of sources to large streams and rivers, rather than the distance of sources to the coastal waters, was an important determinant of nitrogen delivery to the estuary in the Mississippi Basin, and possibly also in other large river basins. A nitrogen-15 stable isotope tracer study of nitrogen dynamics as in head streams of all biomes throughout North America showed that nitrogen uptake length increased significantly with the volume of river discharges. Seasonal variation in nitrogen removal rates has been found in a range of studies. From this, it can be inferred that there are a range of biogeochemical and hydrological controlling factors such as vegetation, nutrient and carbon concentration, temperature, and discharge volume on nutrient removal that are significant and may confound the stream size impact. Ensign also argued that the role of head streams on nutrient retention might be biased by the fact that more sampling and experimental work may have been done in lower rather than higher order streams. More research on nutrient spiraling controlling factors in large rivers and entire river networks would be valuable to estimate nutrient removal from streams and rivers to coastal waters. Dissolved nutrient retention mechanisms in streams and rivers include biological uptake by primary producers, microbial activity such as coupled nitrification, denitrification, and anammox, volatilization, physical adsorption, and sedimentation. Evidence suggests that in lower order streams, the greater dominance of biota attached to substrata results in higher retention of nutrients and intensive nutrient recycling within the attached communities. Removal of the attached microbial communities, as was the case in a desert stream following a flood that eliminated most of the biota, reduced the retention of nitrogen to very low levels, slowed recycling rates, and increased spiraling length. Nitrogen retention increased rapidly as the attached algal, and bacterial community re-established. Riparian zones were found to mitigate in-stream nutrient retention either by changing in-stream ecological features or providing energy inputs into streams. A range of studies showed that natural ecological restoration of streams and rivers can increase nutrient retention. Clear evidence of riparian vegetation assimilating nitrogen-15 isotopes of inorganic nitrogen from a Sonoran desert stream has been reported, suggesting a strong hydrological linkage between the stream and riparian zone in arid areas. The enhanced interaction between sediment and water in aporeic zone can reduce nutrient uptake length and therefore increase nutrient retention. Figure 14-12, conceptual model of dissolved inorganic nitrogen dynamics in headwater stream ecosystem. Ammonium and nitrate enter the stream reach via stream flow and lateral seepage. Ammonium removal is due to uptake by primary producers, bacteria and fungi, plus direct nitrification and physical adsorption to sediment particles followed by sedimentation. Indirect nitrification is the conversion of ammonium mineralized from organic matter to nitrate. Nitrate removal from the water is primarily via assimilation by biota and denitrification on the channel bottom. Regeneration is the release of ammonium and nitrate from the stream bottom back to the water column and is the net result of several interacting processes, including mineralization, indirect nitrification, denitrification, DNRA, anammox, and reuptake by organisms. Nitrate and ammonium remaining in the water are exported downstream. Okay. So we have like three major processes. So one process in red, one process in yellow, which involves biota, and one process in green. All right, so the process in red, we have ammonium and nitrate in seepage from catchment getting to the sediment. We have the stream bottom here. We have ammonium and nitrate in the stream flow. And we have pretty much like this ammonium regeneration. And then part of this ammonium also gets into absorption to sedimentation. However, most of it gets into the assimilatory ammonium uptake by biota. And then we also have a direct nitrification into nitrate, and that nitrate can be also uptaken by the biota. So the biota both works with ammonium and nitrate. Then we have ammonium to nitrate through indirect nitrification. So here we have a direct nitrification process with the biota, and then here we have an indirect nitrification process without, they're not saying really, with biota per se. And then we have mineralization, ammonification through the DNIR process from nitrate to ammonium. And then we have the production of dinitrogen through denitrification of nitrate, and the production of dinitrogen through the NMOX pathway from either nitrate or ammonium. And then we have ammonium and nitrate export outside of the watershed, and then a portion of it gets regenerated. Denitrification and NMOX are important permanent nitrogen removal pathways. A series of nitrogen-15 tracer additions to 72 streams in the United States and Puerto Rico showed that the denitrification accounted for a median of 16% of total nitrate uptake across all streams, and exceeded 43% of total uptake in a quarter of the streams. These data also suggested that the total uptake of nitrate was related to ecosystem photosynthesis, while denitrification was related to ecosystem respiration. Nitrogen retention via NMOX has also been determined in coastal waters, for example, New River estuary and two eutrophic coastal rivers in northeast China. A review study of 79 hydrologically connected streams and rivers showed that restoration of the hydrology of streams and rivers in urban and agricultural watersheds significantly increased nutrient retention. These authors showed that storm characteristic, intensity, times, and size affect hydrological connectivity in intermittent streams and thus affect denitrification and nitrogen retention. The changes in river flow regime as a result of dam construction were also found to change nitrogen retention based on comparison between sites upstream and downstream from dams. One study found equally high variation in nutrient retention when intermittent and constant flow rivers were compared, suggesting high resilience of biological communities responsible for nutrient uptake and or significant control from the local catchment and climate conditions. Subchapter A, dissolved and particulate nitrogen. The concentration and proportion of dissolved inorganic nitrate, nitrite, and ammonium and organic nitrogen compounds in rivers can vary substantially over space and time because of many competing reactions occurring in the nitrogen cycle. For example, high concentration of nitrite can occur in large rivers in summer under warm, slow-moving conditions as a result of disseminatory nitrate reduction in anaerobic sediments. Catchment characteristics such as land use as well as hydrology can play an important role. Higher nitrate and ammonium concentration are found among rivers influenced by anthropogenic activities such as fertilizers use in agricultural land and industrial effluent emissions. So we kind of looked at that actually yesterday when we looked at the pattern of denitrification and nitrogen retention. Maximum nitrate concentrations during storm flows were directly related to the magnitude of the storm, and resulting high discharge were inversely related to the frequency of storm events. A study from 57 European rivers showed that both nitrate concentration and its proportion of total nitrogen increased along a gradient from ultra-oligotrophic to hypertrophic rivers, and that dissolved organic nitrogen was the important secondary constituent of the total nitrogen, even in the most eutrophic rivers. Dissolved organic nitrogen constitutes a major part of the TDN, or the total dissolved nitrogen, concentrations. In subarctic and humid tropical rivers, DON can constitute over 90% of the dissolved nitrogen. Terrestrial vegetation in a riparian zone influences nitrate and ammonium loading from catchments to streams, with nitrogen concentration from runoff generally being higher during periods of vegetation dormancy or following harvesting or fire. The width of the fully vegetated or riparian zone on both sides of the river can be an important indicator of its inorganic nitrogen removal efficiency. Although dissolved inorganic nitrogen concentration and discharge can vary widely on a diurnal basis and seasonally, dissolved organic nitrogen concentration remains relatively constant. Concentration of particulate nitrogen in streams and rivers can increase dramatically during flood events and in catchments with higher rates of soil corrosion and deforestation. Nitrogen cycling. Channel and riparian sediments can absorb a significant amount of ammonium from the water column. The physical absorption of ammonium to sediment particles is dynamically coupled to sources from groundwater ammonification and transformation of dissolved inorganic nitrogen by nitrification, denitrification, and nitrate reduction. The duration of the storage of absorbed ammonium can be highly variable. Some studies indicate that the appreciable retention can occur in summer months and contribute from 12 to 25% of nitrate released subsequently by nitrification in summer. The riparian zone, when water saturated and parafluvial, they're part of the active streams channel without surface water that is connected hydrologically with the surface stream water. And aporeic zones, this is the areas of the stream bed and near stream aquifers through which ammonium and dissolved organic nitrogen to streams. So areas of the stream bed and near stream aquifers through which stream water flows are often major. So let's read this sentence. There are lots of parentheses in this sentence. So let's review the sentence without the parentheses. It's going to be a little simpler to read. The riparian zone, when water saturated and parafluvial, and aporeic zones are often major internal sources of ammonium and dissolved organic nitrogen to streams during base flow conditions. Once within a stream, nitrogen uptake by attached microbes may be the primary mechanism controlling spatial and seasonal variation in the water. This in turn is regulated by local environments, including light, temperature, discharges, and water velocity. However, an ammonium enrichment study in a mountainous tropical river network in Puerto Rico showed that nitrification can rival microbial and algal assimilation of ammonium in the stream water, thus resulting in additional nitrate export from those streams. Reduction in stream nitrate concentration at base flow during the day compared with night suggests that uptake by photoautotrophs can be an important retentive process. This has been shown in cooler seasons when forest vegetation is dormant or deciduous and light levels are relatively high. Sediment denitrification rates in streams may be controlled by hydrologic residence time, nitrate concentrations, and organic carbon quality and quantity. A meta-analysis study showed that the relative availability of carbon and nitrogen can be more important factor limiting denitrification rates. The nitrification rate is likely to be limited by substrate ammonium availability. However, in a study of 18 agricultural and urban streams from the Kalamazoo River catchments, nitrification was found positively related to sediment carbon content, but not to ammonium concentration, probably due to benthic decomposition providing ammonium via mineralization. The processes of nitrification and denitrification often function simultaneously and reciprocally in running water sediments, where many macrozones of steep redox gradients occur in the aporeic zone of the stream bed. The effectiveness of the denitrification and nitrification processes in the aporeic zone of stream sediments depends greatly upon the redox conditions within the interstitial water. Flow conditions within the sediment interstices are quite variable, especially because of localized differences in sediment composition and texture. In organic-rich sediments, the redox profiles can be quite similar to those in lake sediments, in which the zone of nitrification can be restricted to the upper 2 to 3 millimeter, and intensive denitrification occurs below the stratum of nitrifier activity. Denitrification also occurs in shallow riparian sediments connected to the stream by the aporeic zone. The presence of suspended sediments in two large rivers from China, the Yellow and Yangtze rivers, was found to enhance nitrification, coupled nitrification, denitrification, and anamox in rivers by providing both oxic and anoxic microenvironments and larger surface areas for contact between bacteria and nitrogen substrates. And that will be it for today. And tomorrow I'll pick it up with this subsection as compared to lakes and reservoirs. And thanks for attending today, and I'll see you tomorrow. Bye.